Hello, my name is Henry Hardart, and I will be presenting today about the roles of molecular gradients as positional cues both early on and late in development. Asymmetry plays an incredibly important role in development and is a theme that has come up numerous times over the course of this class, whether it takes the form of an egg with an animal and vegetal pole, a neuron with distinct apical and basal structures, asymmetric cell division, or differing concentrations of morphogens at different parts of the body. Asymmetry is a critical feature in a developing organism. In my presentation, we will focus on how these asymmetrical morphogen gradients provide vital positional information during different stages of development. These gradients are incredibly powerful tools. Early on in development, using just a few proteins forming gradients along different axes, individual cells can tell where they are in the body solely based on the concentration of these morphogens. However, as development progresses, neurons require increasingly precise and specific spatial information for processes such as axon guidance and map formation that require different positional cues. This leads to one set of molecular gradients being used early on in development that provide general positional cues for cell differentiation and gene expression, while another set of molecular gradients that are established later in development provide more granular detail for more precise tasks. One example of early molecular gradients that provide positional cues is the Wnt gradient. Wnt is a posteriorizing factor that, along with other morphogens such as FGF8 and retinoic acid, define the early anterior-posterior axis. These morphogens are soluble factors that are secreted from either the anterior or posterior poles of the developing neural tube and cause cells along this axis to adopt different cell fates. To demonstrate how these morphogen gradients are able to have such profound effects on cell fate, I've included some additional information about the Wnt signaling pathway. In the absence of Wnt, the transcription factor beta-catenin is normally phosphorylated and tagged for degradation by the kinase GSK3. However, when Wnt interacts with the frizzled receptor, this receptor recruits some of the proteins that are required for GSK3 to function, such as axin, thus inhibiting the GSK3 kinase. This prevents beta-catenin from being degraded and allows for the targets of beta-catenin to be expressed. A combination of this signaling pathway and the signaling of various other morphogens along the anterior-posterior axis lead to the differential expression of transcription factors and proteins along the axis, and therefore different cell fates along the anterior-posterior axis. Another morphogen, sonic hedgehog, is secreted by the floor plate of the neural tube and also forms a gradient along the dorsal ventral axis, inducing changes in cell fate such as the dorsal horn of the spinal cord housing sensory neurons, while the ventral horn contains motor neurons. These morphogen gradients act together over long distances and provide a general coordinate grid that spans the entire body plan, so that cells can develop differently based on their position on both the anterior-posterior and dorsal-ventral axes. However, later on in development, especially in the CNS, this singular grid is not specific enough, because there are increasingly complex and detailed structures that have to develop. This necessitates the development of secondary organizing centers in the CNS to provide more detailed positional information. Secondary organizing centers emerge later on in development to provide additional positional cues when creating the detailed structures such as the brain. These secondary organizing centers emerge directly from these early molecular gradients that we have just discussed. For example, at the intersection of Wnt and similar posterior molecular systems and their respective inhibitors, expression of a transcription factor engrailed is induced, and the isthmic organizing center emerges. These later organizing centers are responsible for the local organization of nearby tissue. The isthmic organizer, for example, secretes FGF8 and is responsible for the induction of the cerebellum and midbrain. This facilitates the development of more complex structures in the central nervous system. Another of the large challenges of development is the creation of remarkably precise topographic neural maps. One example of these maps is the retinotopic map. The retinotopic map is a representation of your visual field in your visual cortex, where neurons that are spatially close to each other in the brain respond to visual stimuli that are next to each other in your visual field. Creating this map requires more precision than gradients of soluble morphogens are able to provide. To overcome this problem, gradients of surface proteins called ephrins are expressed in the tectum and visual cortex, and gradients of ephrin receptors are expressed in retinal ganglion cells, allowing for a tremendously detailed map. Ephrin surface proteins provide a repulsive cue to axons that are expressing the ephrin receptor, so retinal ganglion cells that highly express the ephrin receptor go to parts of the tectum that do not express ephrin, while ganglion cells that do not express the ephrin receptor are routed to parts of the tectum with high expression of ephrin. Similarly, the auditory system creates a tonotopic map that groups neurons that respond to similar frequencies together in the auditory cortex. Interestingly, ephrins have also been implicated in the formation of this map. This is yet another example of how precise molecular gradients play an important role later in development, and also demonstrates interesting parallels between the development of different sensory maps on the cortex. In conclusion, as development progresses, the molecular gradients providing positional cues progress from more general cues that provide information about location within the entire body plan to very specific cues that provide information about where one is within the brain or even within one specific part of the cortex. Thank you very much, and I hope you enjoyed my presentation.